Life Sciences, Week 4, Video Lecture 8, Organic Compounds and Lipids. All right, guys, so we're continuing our journey through some of the more common organic compounds we have in the body. And so now we're going to take a look at lipids. So our objectives here are to describe the structure and function of triglycerides, phospholipids, and steroids, and describe the differences. Let's see, structure and functions. Yeah, that's what we're doing. So that means we'll look at what these molecules look like, then we'll also take a look at their functions in the body. We'll describe the differences in structure and characteristics between saturated, monounsaturated, and polyunsaturated fats, and we'll explain how emulsifiers work. So here we go. All right, these are sort of two major categories of lipid molecules that we're going to look at. On the left, this is a triglyceride, and we're going to start looking at those. And then after we look at triglycerides, we're going to look at the molecule on the right here is cholesterol, which is built off of a steroid foundation. So it's one of the steroid molecules. So we'll be looking at steroids, and we'll be looking at triglycerides and variations on the triglyceride theme. So starting with triglycerides. So triglycerides have a basic backbone to them, which is called glycerol. And the glycerol molecule is 3 carbons long. So we can see these 3 carbons across the middle here. And so what this is showing is just glycerol all by itself. And so what we see is the 3 carbons. Each of the 3 carbons has hydrogens attached to them where they're not attached to each other, or where they're not attached to an oxygen. And that oxygen has a hydrogen attached to it. So once we put a triglyceride all the way together, it's going to look a little different. But this is the basic backbone that we're going to find in any triglyceride. So let's see. I'm just looking at the notes here and seeing what else I should say before we dive all the way into triglycerides. It looks like we're good. Okay, so triglycerides. Let's go on. So triglycerides, if we consider the name triglyceride, we can hear this glycerol part of it, right? That's the glyceride part. Where does the tri part come from? The tri part comes from 3, it means 3, and what there are 3 of in a triglyceride are 3 fatty acid chains. So we're going to take a look at the fatty acids in more detail in just a bit, but first let's take a look at how the fatty acids attach to the glycerol molecule. So on the far left here, we're looking at a glycerol molecule. We've rotated it 90 degrees from what we were looking at on the previous screen. So now we see the 3 carbons all, one above each other, each of, whatever, they're standing on each other's heads. So that's on the far left. So if you can see those 3 carbons, and we see the hydrogens attached to them. They're just showing super long bonds between those carbons, just so there's space to show the rest of it. And then we see the OH on the other side of the carbon. So we also see that OH is circled along with a hydrogen molecule of something else. So that something else is a fatty acid. And like I said, we'll get into that. But let's first look at how the fatty acid attaches to the glycerol molecule. So at this point, you might have guessed already that there is, here they're calling it condensation, but it's what we've been calling a dehydration synthesis reaction. So we're going to take that circled oxygen and 2 hydrogens out and make a water molecule out of it, and replace it with a bond. So there'll just be a bond between the remaining oxygen that is on the end of the fatty acid and the carbon of the glycerol molecule. So we can see that happened in each of those 3 places. So that means in order to build a triglyceride, we need to have a glycerol molecule, and we need to have fatty acids, and then we can do that dehydration synthesis reaction and attach the fatty acids to the glycerol molecule. Now, of course, we can run that reaction the other direction, and we can separate the fatty acids off of the glycerol by doing hydrolysis. So if we insert a water molecule in there, then we'll run the reaction the other way, and we'll have separated the glycerol molecule from the fatty acids. Okay, so that's how we attach them to each other. Let's take a look at the fatty acids in a little more detail. So fatty acids are not called fatty acids just because they like the term fatty acids. As you probably have guessed, they're called fatty acids because they're acids. So what about them is an acid? So remember, of course, that an acid is something that when we put it in water, it's going to donate hydrogen ions to that solution. And so what we're looking at here is a carboxyl group, and we've seen carboxyl groups before. We've seen them in amino acids. So if you recall, an amino acid has a central carbon. On one side it has an amino group, that's nitrogen and 2 hydrogens, and on the other end it has the carboxyl group, which is a carbon and 2 oxygens and a hydrogen. And that is what we're looking at here. So what's in blue here is the carboxyl group, the carbon, 2 oxygens, and hydrogen. And then what they're showing as an R is whatever that carboxyl group is attached to. So in an amino acid, the carboxyl group is that R would be the central carbon of the amino acid. In a fatty acid, that carboxyl group, oops, I'm skipping ahead here. In a fatty acid, the carboxyl group attaches to one end of a chain of carbons. So that chain of carbons can look a little different depending on what type of fatty acid it is. So there are 3 types or 3 categories of fatty acids. We have saturated fats, monounsaturated fats, and polyunsaturated fats. So you've probably heard of these before. And so let's take a look at where these names come from and how the fatty acids look different depending on which of these categories they fall into. So first we'll look at saturated. But let me just mention, since it's on here, the monounsaturated fatty acids are also called MUFAs. So that stands for, the MU is monounsaturated, and the FA is fatty acid. So monounsaturated fatty acid is a MUFA, and a PUFA is a polyunsaturated, that's the PU. And the FA is the fatty acid. So maybe, sometimes when you're reading labels, you see MUFA, like especially if something like is some sort of a supplement that supplements good fats or something, you might see MUFA on there. Anyway, so let's take a look starting at saturated fats. So saturated fats, I would love for you to do your best to set aside any thing that any prejudice you might have against saturated fats. Okay, so here we're looking at them totally neutrally, just as a molecule, but I especially am asking that of you because saturated fats are really fantastic for us. They're not at all detrimental to fantastic, vibrant health in the slightest. Okay, so saturated fats. What makes them saturated? Why are they called saturated? So in saturated fatty acids, what we see is, so first of all, what we're looking at is on the left side here, we see the carboxyl group, right? That carboxyl group that we looked at, the C, the OO, and the H. And that's that carboxyl group that can donate hydrogen ions to a solution that makes fatty acids acids. Okay, so it's that hydrogen that comes off. That's what we can observe happening, and that's where the acid part of fatty acid name came from. So the rest of this is a chain of carbons, all covalently bonded to each other, and you see that there are single covalent bonds between every carbon, and every carbon has 4 bonds made, so 2 with each carbon, except for the end one, and 2 with 2 hydrogen atoms. Okay, so the term saturated fats comes from the fact that this molecule is saturated with hydrogen. We couldn't get any more hydrogen in here if we possibly tried, because we haven't, and so which means this is very stable because all of these carbons have a single covalent bond in the 4 positions that they want to have that, and so it makes it very stable. Nobody here, none of these atoms are interested in entering in, well, except for the carboxyl group. The rest of it is not really interested in entering into chemical reactions. It's quite stable and happy as molecules go. So qualities of saturated fats. If you had a lump of saturated fats or fatty acids that looked exactly like this, and you had them all together in a pile on your kitchen counter, here's some things that they would look like. Number 1, they would be solid at room temperature. And they would be more solid if you stuck them in the refrigerator. So for instance, like lard or butter are not exclusively made of saturated fats, but they have a lot of saturated fats in them. And like coconut oil or palm oil have a lot of saturated fats. So, so if they're in the refrigerator, they're rock hard. If they're on the counter and it's not summer in Tucson, they're still solid, okay? So part of what that tells us, that basically speaks to like the boiling point, or the point at which these molecules begin to break down. And kind of change how they're presenting. So essentially they are solid at room temperature, and which means it takes kind of a lot of temperature, a lot of heat to sort of rock their molecular boats and get them moving around enough to turn them into a liquid. Which means they're quite stable molecules. You can expose them to heat, you can expose them to light, and it's not going to affect them unless you expose them to a lot of heat and a lot of light. That's saturated fats. Okay, next type are monounsaturated fats. So again, on the left we see the carboxyl group. It's represented differently than on the previous slide. And so here we can see the actual bonds between each of the atoms of the carboxyl group. And then to the left of the carboxyl group we see a chain of carbons with a whole bunch of hydrogens attached. So the difference between this one and the previous one is that we don't have single covalent bonds all the way along. There's one place where we have a double covalent bond. And so if we count carbons, starting with the carboxyl carbon, 1, 2, 3, 4, 5, 6, 7, 8. So between 7 and 8, those two carbons, that's where we're seeing that double covalent bond. So those two carbons are sharing 2 pairs of electrons, whereas all the rest of them are just sharing 1. So a monounsaturated fat, so it's kind of, we would think that a double covalent bond is stronger than a single covalent bond, but it's not really. It's actually kind of a wobbly point in this long molecule. And so if this molecule is exposed to too much light or heat and is going to denature some, that is potentially one place where it's going to break down. Potentially hydrogens might jump ship too. But anyway, so monounsaturated fat is a fatty acid in which there is one place that is not completely saturated with hydrogens, and that's at that place where there's that double bond. So mono means one, un means not, and saturated means saturated with hydrogen atoms. So a monounsaturated fat has one position in which the carbons are not completely saturated with hydrogen, and we can see that that's where that double bond is. That's a monounsaturated fat. The third kind is polyunsaturated. So you've probably guessed by now that similar to a monounsaturated fat, it has some place where it's not completely saturated with hydrogen. Therefore, it has some place where there's a double covalent bond between the carbons. But since this is called a polyunsaturated fat, there's more than one place where that's going on. So we're looking specifically at linoleic acid, which is an 18 carbon long fatty acid. And it actually has 3 places where there are double covalent bonds. So there are 3 positions where those carbons are not fully saturated with hydrogens. So that's a polyunsaturated fats. Oh, I didn't mention about other characteristics. So monounsaturated fats are solid at colder temperatures. So monounsaturated fats, for instance, are abundant in olive oil. Okay, that's one of the great health benefits of olive oil is the monounsaturated fat, oleic acid that's in it. So monounsaturated fat, so imagine you have a bottle of olive oil. If you put it in the refrigerator, it gets solid pretty quickly and easily. So much so that if you take it out, you have to wait quite a while for it to be pourable. But if you leave it on the counter, unlike lard, olive oil will be liquid at room temperature. Okay, so monounsaturated fats will turn liquid at a cooler temperature than saturated fats will. Polyunsaturated fats are liquid at even a cooler temperature. So if you had a bottle of polyunsaturated fats, so polyunsaturated fats come from seeds and grains. So you could have corn oil or peanut oil or something like that, something that comes from a grain or a seed or a nut, and that would be rich with polyunsaturated fats. If you had that on the counter, it would be liquid. If you put it in the refrigerator, it would still be liquid. So you have to get much colder temperatures to turn that into a solid. So these guys stay liquid. And you can kind of relate then that the number of covalent bonds in it relates to how wiggly the molecules are, and so how much colder you need to make it solid, right? So if it's solid, these things are moving around less. And but if they're liquid, they're moving around more. So these guys are more apt to move around, and again, it's that double covalent bond that gives them sort of that wiggle, that makes them, liquid at cooler temperatures, but also makes them slightly vulnerable. So polyunsaturated fats can start to denature and fall apart with exposure to light and exposure to heat. So, and monounsaturated fats can too. So, which is why when you're buying olive oil, very often it comes in a green bottle. So they put it in a green bottle because they're trying to keep light from interacting with the molecules so that they don't start to decay there in the bottle. Polyunsaturated fats, they don't usually sell in a sometimes they do sell it in a green bottle, but a lot of times they don't, and it's partly because they're already fairly broken down, because it doesn't take a lot of heat or light exposure to start to break these molecules down, which means these guys are ones that don't like to be used for like high temperature cooking, because they'll just fall apart and smoke quickly, and it depends. You can stabilize them. There are things you can do, and a lot of times we do use these for cooking, but that can create some problems that we're not going to go into right now. So let me just point out on the bottom, we have a depiction, sort of a shorthand depiction of this same molecule that's shown up above. So up above, we see all the carbons and all the hydrogens. Down below, we see sort of that shorthand version where each of the zigzags, at every corner there's a carbon. A single line means that there's a single covalent bond. It's implied that there are hydrogens filling in the rest of the bonds with the carbons, and where there's a double bond, there's a double line, there's a double covalent bond. So another thing about fatty acids, well, and we've talked about this with in organic compounds in general, when there's a double bond, the molecule will basically have a little turn or a little sort of corner in it that is sort of depicted in this top version here. And when that happens, it can either go one way or it can go the other way. So, so in organic chemistry, those terms are cis and trans. So we can kind of think of them as sort of right-handed and left-handed. So, we could have this exact same molecule that we're looking at with the same number of carbons and double bonds in the same locations. But if where this one has a cis bond in it, in other words, say it's a right-handed turn, and if another one had a trans, say a left-handed turn in that same location, the body would perceive that as a completely different molecule. And if you're getting ahead of me, then you've probably already guessed that is where the term trans fats come from. Most of our naturally occurring fats are cis fats. And then sometimes when we make fats in the lab, we put the right number of carbons and hydrogens all together, but we can't control necessarily whether it turns cis, whether they come together as a cis molecule or a trans molecule. So trans fats are just sort of, I guess, mirror images of cis fats, and cis fats are mirror images of trans fats. Some cis fats are totally naturally occurring and just fine, but some trans fats are naturally occurring and just fine, but most trans fats are ones that are sort of a laboratory attempt at making a cis fat that didn't turn cis, and then when we put that in our body, our liver's like, what the heck is this? I can't even identify it. I don't know what it is. This is strange. And so it just will kind of store it away because it can't use it. It doesn't know what to do with it. So those are trans fats. Anyway, that's not in your notes. It's a total digression. Let's move on. Okay, let's see here. What else? Let me scroll to where we are in the notes. So we talked about saturated fats, we talked about monounsaturated fats, we talked about polyunsaturated fats. So what we're looking at here is just a chart that shows that shorthand of a number of different common fatty acids. So we have stearic acid on top. It's showing that it's an 18 carbon saturated fat. So we can tell it's saturated just by looking at it, because all of those bonds in between the carbons are single covalent bonds. Oleic acid, this is the one we mentioned that we were looking at before. It's a monounsaturated fat, so it has one place where there's a double covalent bond, and it's also 18 carbons long. And linoleic acid is the next one here. It is an omega-6 fatty acid. That's that N6 that we see there. That omega-6 refers to that the first double covalent bond is in the number 6. It's on the number 6 carbon. It's in that position. That's where that 6 term comes from. But it's 18 carbons long, and it has 2 places where it has double covalent bonds. And so if you look at this nomenclature on the left, you can see the names of each of these fatty acids. So stearic acid has 18, and then has a colon, it has a 0. So 18 means it's 18 carbons long, and then that 0 means there are no double bonds. Oleic acid is 18 carbons long, it has the colon, and then it has a 1. That 1 means there's 1 double covalent bond, and then it has an N9, which really means omega 9, and that means that that double bond is at the number 9 carbon. So we call that an omega 9 fatty acid. Linoleic is 18 carbons long. It has two double covalent bonds. The first one is in the number 6 carbon position, so this is omega 6 polyunsaturated fatty acid, and then the bottom one is alpha linolenic acid, which is 18 carbons long. It has 3 places where it has double bonds, so that's the 18:3, and then it has N3 there, which means it's an omega 3 fatty acid, which means the first double covalent bond is on the number 3 carbon. Okay, so and it's showing that those are both essential fatty acids, which means just like with amino acids, if they're essential fatty acids, our body cannot manufacture them. We need to take them in through the food we eat. All right, so now if we put it all together, we have a triglyceride. So again, here on the left, we have standing upright, we have those 3 carbons that form the glycerol backbone of the triglyceride. Each of those carbons has a carboxyl group attached to it, and then attached to that are the fatty acid chains. And so one of the things we're seeing here is that the fatty acid chains in a triglyceride do not all have to be the same fatty acids. So here we're seeing the top 2 fatty acids are saturated fatty acids, and the bottom one is monounsaturated. So that's fine for a triglyceride. Sometimes they're all 3 the same fatty acids, sometimes there's 2 different ones, sometimes there's 3 different fatty acids making up a triglyceride. It just totally depends on where it came from, and what molecule made it for what reason, and all that. So what are triglycerides used for in the body? Triglycerides are the fats that we use as stored energy and for heat insulation. So on the top of page 6 in the notes, we have the uses for triglycerides in the body. So I think I don't have it here. No, I don't. Okay, so triglycerides in the body provide heat insulation. So these are, again, the triglycerides that are stored in adipose tissue. So they provide heat insulation, they provide a physical padding for our body. They also provide an electrical insulation. So these triglycerides are the fats that we're going to find in the cells that wrap around our neurons that provide an electrical insulator to them, or for them. And then, but basically our triglycerides are our stored energy in our body. So triglycerides can store a lot more energy in their bonds than carbohydrates can. So that makes sense for triglycerides to be our long-term stored fuel source, because we can store a lot of fuel in a small package. Whereas carbohydrates, we can't store as much potential fuel in the bonds of carbohydrates, but we can get to it quicker and easier. So carbohydrates convert to ATP more quickly and easily than lipids do, than triglycerides do. So it makes carbohydrates, and remember our stored form of a carbohydrate is glycogen. So our glycogen is our stored quick access stored energy, whereas our adipose, or our triglycerides, are longer term stored energy. Okay, so that's what we use triglycerides for in the body. Now let's look at phospholipids. So phospholipids have essentially the exact same structure as triglycerides, except they don't have 3 fatty acid chains, they only have 2. So if we look at this diagram, the one on the left, so there's a red circle that's labeled hydrophilic head. So right across the middle of that circle, we see CH2, CH, CH2. That is the glycerol backbone of this molecule. Below the first 2 carbons, we see the carboxyl group. So we see an oxygen, a carbon, and then double bond to an oxygen. So that's the carboxyl group. And then below each of the carboxyl groups, we see fatty acids. So one of the fatty acids is a saturated fatty acid, and the other one is an unsaturated or a monounsaturated fatty acid. Okay, so that's that. That is all very familiar. It looks just like a triglyceride, right? Now let's look at that third carbon. So attached to the third carbon and flipped around to the other side of the molecule, we have a phosphate group. So the phosphate group is in its own little circle there. So it's a phosphorus and 4 oxygens attached to it, and then also another side group attached to that. So up on top we see an R, and that represents another side group. So the phosphate portion of the molecule has a negative charge to it. That other side group part of it will have a positive charge to it. And so what that means is we have a polar aspect to this molecule. So remember that polar covalent bonds are bonds in which the electrons are not equally shared, and it gives rise to a slightly negative and a slightly positive portion of the molecule. And so that's what we have in this hydrophilic head of this molecule, is a polar component to it. So there's a slightly negative part, that's the phosphate part, and a slightly positive part, that's that R side group there. So having a polar aspect to a lipid molecule means that polar aspect can interact with water, because water has polar covalent bonds in it, as we know. The oxygen part of water tends to be more negative. The hydrogen part of water tends to be more positive. So those will attract to this slightly positive and slightly negative aspect of this phospholipid. And so these are a few ways that we can depict a phospholipid. So we can see the structural formula here on the left. That middle diagram shows all of the carbons and the hydrogens and the oxygens and the phosphate, etc. And then we've zoomed out to take a look at this molecule from a little more of a distance in that third diagram, where we just are seeing what we call the hydrophilic head. So the hydrophilic head is the part with the polar aspect to it that can interact with water. And then the hydrophobic tails, they're hydrophobic because they're fatty acids, and we know that fats stay away from water whenever they can. They don't like to hang out in water. So they're hydrophobic, the head is hydrophilic, and that's what a phospholipid looks like. So those qualities give phospholipids unique characteristics that we utilize structurally in cells and in our body. So what we're looking at here is a phospholipid bilayer. So phospholipids will arrange themselves so that all of the hydrophilic tails, in other words, hydrophobic tails, so in other words, all of the fatty acids will orient toward each other, because they're, in essence, they're like a fat droplet. They want to stay out of water. Whereas the hydrophilic heads are happy to be in water. They interact just fine with water. The water molecules, the slightly negative and slightly positive aspects of water molecules, connect with the the charges that are opposite of that hydrophilic head in a phospholipid. So that makes these guys the perfect thing to use to separate. If we get a whole big sheet of these together, we can separate 2 water compartments from each other. And let's take a look at what that looks like. So what we're looking at here is a portion of a cell membrane. So you can see all the phospholipids with their red hydrophilic heads and their yellowish hydrophobic tails, all oriented to keep the tails out of the water and the heads in the water. And so if the top part of this is the extracellular fluid and the bottom part of this diagram is the intracellular fluid, then everybody's happy, right? Because we have a nice division between the outside of the cell and the inside of the cell. That's a really solid division, because things, that are water soluble can't seep in through those in between the phospholipids, and but the phospholipids are happy because their hydrophobic parts are away from water and their hydrophilic parts are in water. But the other thing that we see here is we're starting to put together all of these organic molecules that we've looked at. So we haven't looked at cholesterol yet, but we're about to. We do see cholesterol on here, but we see some various proteins. So we see some proteins inside the cell. They're called cytoskeletal filaments on here. We see proteins within the cell membrane. So we see one labeled as a protein channel. We see some labeled as integral membrane proteins. So they're different proteins. And some of these proteins we can see are smaller proteins that just have their secondary structure to them. So they're just like spirals, but some of them are more complex than that. And we see some carbohydrates here. So up on top, we see something that's labeled a glycolipid. That means that there's, well, let's see, a glycolipid. Yeah, it's a lipid with a carbohydrate attached. So those green hexagons, or well, maybe they're just circles, but whatever they are, those are carbohydrate molecules. And maybe they're acting as receptors or maybe cell identity markers. So we've seen some of that, some of those functions, some of those structural functions for carbohydrates in the body. And we also see glycoproteins too. So we see proteins with carbohydrates attached to them. Again, identifying that, or maybe it's acting as a receptor or something like that. Okay, so this is, so here are three of these molecules that we've looked at coming together in real specific ways to form a bigger unit with functions within that bigger unit. And phospholipids, we can make cell membranes out of phospholipids, which is what we were looking at, but we can also make membranes for other things. So basically one of the things that we're looking at here is on the right, on the bottom, is if we imagine that cell membrane bigger and bigger and bigger until it turns into a sphere, that's kind of what we're looking at here. So that's the idea of a cell membrane. But we also can, now imagine if we had like a single layer of phospholipids instead of a double layer, the way we do for cell membranes. A single layer of phospholipids can form a nice carrier for fat molecules. So if we have all of our hydrophobic heads on the outside where the water is and our hydrophilic tails on the inside, did I just say that wrong? The hydrophilic heads on the outside, the hydrophobic tails on the inside, other fats could hang out. So imagine that in a sphere like that, but the sphere would be filled with fats where the hydrophilic tails want to be. And so imagine that as a transport vesicle. So we could transport fats around the bloodstream in such a vesicle that's made out of phospholipids like that. Okay, let's move on and take a look at steroids. So steroids are the other group of lipids that we're looking at, and we can see just here that steroids have a very different structure to them than triglycerides do. So steroids, so this diagram on the left shows the basic structure of a steroid molecule. So steroids are, at their foundation, are made up of 17 17 carbon atoms that come together into, that form four, what are these called? Rings. So the rings are labeled A, B, C, and D. We can see A, B, and C are hexagon shaped, D is a pentagon shape. So all steroids have that exact same starting point. And then on the right, what we see is cholesterol. So a cholesterol molecule is an example of a steroid, and we can see that to the number 17 carbon, it's not labeled that on here, but I'm just telling you it's the number 17 carbon that this side group up on top is attached to, and is that side group and these other attachments that have taken this steroid backbone and turned it into a specific molecule that we'll call cholesterol. And cholesterol is kind of a good, it's another starting point molecule that we use to make all sorts of hormones and things in the body. So let's take a look. So that's the basic structure of a steroid. So just at first glance, we can tell that steroids, they sort of look a bit like carbohydrates in a way, right? Because they have all those rings to them the way carbohydrates do. but they're not the same as carbohydrates because they don't have the amount of oxygen in them that carbohydrates have. In fact, they have hardly any oxygen, depending on what the side group is. There's hardly any oxygen in them. But steroids do have, many steroids do have the ability, though, to interact with water and to interact with lipids. They are lipid molecules. They're lipid soluble, so they can hang out with lipids, but many of them, can have a polar part of it, and they can also hang out with water. So let's look at some examples of some steroid hormones. So again, looking at these, we can see that basic backbone, and there's some variation to it here and there, but we can see this basic 17 carbon backbone that is the steroid component of all of these different hormones. And then we can also see that there are differences between each of these hormones. And so, many of these are hormones that we probably have heard of, so especially the top two and the bottom two, I'm sure we've all heard of. So aldosterone and cortisol are two hormones that come from the adrenal glands, and those are both hormones that are in that category of stress hormones. So a lot of our adrenal hormones are steroid hormones. In other words, they're fat-soluble hormones with a steroid foundation to them. And the bottom two, testosterone and progesterone, are both sex hormones. And so those and other sex hormones, like the one above testosterone, all of our sex hormones also have a steroid base to them. And then, so a whole lot of these hormones, we can turn one into another just by changing the side group on them, or making slight changes to the steroid foundation to it. So anyway, these are some examples. So that kind of tells us some of what steroids are doing in the body. A whole lot of what they're doing is they're acting as signalers, or communication. Basically, they're hormones. A whole lot of our steroids in our body are hormones. And that term steroid, outside of studying organic chemistry, when we hear steroid, we think, oh, that's something that somebody takes if they want to build muscles. They'll take anabolic steroids. Well, what does that mean? That means they're taking hormones that promote growth. It's a specific hormone or a group of hormones that promote muscle growth. And so here are some categories of hormones and functions that those within those categories. So cholesterol is a single molecule. It's not a category, but it's a single molecule that's a starting point of a whole bunch of different things in the body. Vitamin D is one example, but a whole lot of our hormones have cholesterol as a starting point for them. And so sex hormones are a category of steroids. So all of our sex hormones are some type of steroid hormone, so they all have that 17 carbon 4-ring backbone to them. Our bile salts, like cholesterol, both of those things are found in bile, and they have emulsifying qualities, which means those are molecules. We'll talk about emulsifiers here, but this is a sneak peek, I guess, that bile salts and cholesterol both have the quality of they are fats, and so they can hang out with fats, but they also have a polar aspect of the molecule, so they can hang out in water too. So they can sort of bridge the fat and water world of those two worlds of the body. Cholesterol and other stress hormones are also steroid hormones. So cholesterol, aldosterone, and many of the other hormones that come from the adrenal glands. Okay, so those are some functions of steroids. So emulsifiers, what I was starting to talk about on the previous slide. Emulsifiers are molecules that can interact with both water and lipids and can therefore act as a bridge between them. So what does that look like? So this is, I think this is ethanol acting as an emulsifier here. So on the right of this diagram, we see water. Okay, so we see the oxygen, we see the two hydrogens, that's water. And we know that there's a polar covalent bond there, so the oxygen is slightly negative and the hydrogens are slightly positive. On the left, we see what I think is ethanol, and we see that the positively charged, or the slightly positive hydrogen end of water is attracted to the slightly negative oxygen of this molecule. And so a hydrogen bond forms between there. But if we look at the rest of this molecule, we see carbon after carbon with all these hydrogens that looks like a tiny segment of a fatty acid chain, right? And so so we can imagine that that part is going to act like a fat, whereas this part with the act like a fat, meaning fat soluble, can interact with fats, can be with fats, whereas the oxygen part that has a slightly negative charge to it, and the hydrogen attached to it that has a slightly positive charge, will interact with water and will form hydrogen bonds with the water. Okay, so that's what an emulsifier does. It has a part that can form hydrogen bonds with the water, but it also has a part that that bonds to or connects with, or at least can hang out with, fats. And so this is the kind of setup that we get with an emulsifier. So these yellowish circles are droplets of oil, but we have an emulsifier, and the oil is in water, but we have an emulsifier in here too. So the emulsifier has arranged itself, and you can tell it looks sort of like a phospholipid because it has those heads, and here they're showing as white circles. Those heads are hydrophilic, so they are arranging themselves, so they're sticking into the water, but the tails are hydrophobic, so they're hanging out with the fats. But so the result here is that these droplets of oil are all surrounded by the emulsifier, and the emulsifier then, you know, kind of says to the oil, come on guys, the water's fine, let's get in the water. So it allows the fat molecules to distribute more evenly throughout the water, but they can only do that by being surrounded by the emulsifier. So speaking of that, let's take a look at, make sure we covered our objectives. We looked at the structure and the function of triglycerides, phospholipids, and steroids. We looked at differences between saturated, monounsaturated, and polyunsaturated fats, and we explained how emulsifiers work. So that leads us to your homework, which is this: to pour some water into a clear glass or onto a plate, kind of like this, and add an equal amount of oil, or maybe less oil, but some oil. So this is not super precise, and I'm sure Galileo would not like the way we're doing this because I'm not giving you precise measurements, but that's okay, we'll roll with it. Mix the two and observe what happens. Do they mix and stay mixed, or do they separate? Now we're going to add an egg or some vinegar, or you might have something else in the kitchen that you could add, or whatever around. But these are two things that I'm guessing you have around that can serve this purpose. And we're going to mix it again, and we're going to observe what happens. And so all along the way, I'd like you to either take pictures, or you can describe to me what's happening in each step, and then answer these questions. Number one, did the oil and water mix and stay mixed when it was just oil and water? Number two, did the combination stay mixed once you added the egg or added vinegar? And then number three, why or why not? So what is going on here? So I want you to send pictures and responses to the questions to my email, jprovidence@asum.edu. And that's it for this. We have one more video for week 4, which is about DNA and RNA, or nucleic acids, and about ATP, which is a molecule we've been referring to left and right here. And so we'll take a look at that guy. And then that wraps up our week 4, and that means that I can set you free on test number 2. So that is coming up very soon. All right, I hope you enjoyed this video, and I'm sure you're looking forward to the next one. I'll see you in class.